Hello, my name is Jane Suan Wu. I am making this video as a submission to the University of Pennsylvania's inaugural BIN Talks as part of the Benjamin Franklin Scholars Program. My project, currently titled The Effect of Metal Cations on Ptaquiloside Hydrolysis, was conducted at a lab in the Department of Plant and Environmental Sciences at the University of Copenhagen in Copenhagen, Denmark. My supervising professor was Professor Hans Christian Bruun Hansen, and my supervising PhD fellow was Frederik Clauson-Kaas. The lab generously provided me with the training and lab materials required to pursue this experiment. This project sought to analyze the degradation rate of a chemical called Ptaquiloside, PTA, which is a bracken fern-derived carcinogen, in the presence of various dissolved metal cations. Now, this sounds pretty complicated, so what does it mean? Before delving into the experimental process, I shall give some background on why this research matters. The easiest starting point is to understand what bracken fern is and why we care. If you read the Wikipedia description on bracken, you will find that bracken fern is a very durable plant that can outcompete many ground cover plants. It thrives in a large range of soil pHs from the high 2s to the mid 8s. It is found on every continent in the world minus Antarctica. Global climate changes are expected to be relatively favorable to this invasive and fast colonizing plant. One of the several problems with this plant is that it contains toxins that can negatively affect the health of domesticated animals such as cows and the humans that consume it directly, as is a custom in some areas of Asia. Not so great when bracken population is increasing each year. The most critical of the toxins in bracken fern is the carcinogen Ptaquiloside. Several studies in South American countries have shown that cattle that eat bracken can carry a portion of the Ptaquiloside in their milk, which once consumed by humans can lead to increased levels of gastrointestinal cancers. Consumption is the main exposure pathway of concern for Ptaquiloside. This risk is relatively well studied in the case of direct consumption of bracken and consumption of milk in bracken-exposed cattle, but the risk is less studied in the case of groundwater exposure pathways. This risk is one of the main focuses of study at the lab that I worked in at the University of Copenhagen. So, now that we know why bracken and Ptaquiloside matter, we can begin to talk about specific research that has been done on Ptaquiloside behavior. We know that Ptaquiloside degrades over time to turn into multiple other substances, the main one being terrazone B, which is much less risky to deal with, which is good. This is what we want to happen. Research by Al Lewis and several other scientists published in 2006 show that low temperatures around 4 degrees Celsius and slightly acidic pH at around 5.5 have a strong stabilizing effect on Ptaquiloside in aqueous solutions. At higher temperatures and lower or higher pHs than these values, Ptaquiloside degrades at a much faster rate, which is good. This can shorten the time frame for degradation from a week or two to just a few days. Research by Hansen and others, published in 2008, showed that in soil solutions, clay particles can stabilize Ptaquiloside, which is not good, and microbial activity can increase the rate at which Ptaquiloside breaks down, which is good. The experiment that I completed over the course of the summer sought to expand on the previous knowledge of what determines Ptaquiloside stability. The summer before I conducted my experiments, another student at the University of Copenhagen lab by the name of Aline Rousseau conducted an experiment similar to mine, in which she observed the degradation rate of Ptaquiloside in the presence of select metal cations at a pH of 5.5. She used cation concentrations of 0.5 and 5 millimolar in her experiments. She found that at these concentrations, light metals such as sodium, potassium, magnesium, and calcium have no effect on Ptaquiloside degradation rates, while iron and aluminum can increase degradation rates. Copper had only an effect over a limited range of concentrations, and manganese only had an effect in the presence of citric acid buffer, but not MES buffer. The concentrations used in Aline Rousseau's experiment for both metal cations and buffers were very high compared to what would be found commonly in Danish groundwater. The concentrations of the 5 metal cations I eventually chose to study in my experiment were chosen to be much closer to the concentrations found in nature. I chose to study iron 2, copper 2, manganese 2, and nickel 2 concentrations based on an iron 2 content of 5 mg per liter that can be found in Danish groundwater, or the equivalent of 0.0895 millimolar. For magnesium, this was an exception, as there are much higher levels of magnesium in Danish groundwater than any of the other cations under study, and a Leans experiment showed that magnesium at concentrations of 0.5 millimolar had no effect on the degradation rate of PTA. The equivalent of 76 milligrams per liter, or 3.1 millimolar magnesium, was used in the experiment. Samples for the experiment were prepared in a glove box, a device made to prevent the exposure of oxygen to samples, as the cations whose effect I was trying to study were all in their unoxidized states. I chose to conduct my experiment at around pH 7.3, 7.5, to decrease study time needed for the experiment to occur to what I hope would be less than 2 weeks. HEPES, H-E-P-E-S, buffer, instead of MES or citric acid, was used in my pilot stage of the experiment, as this buffer is good for pH in the mid-7s. Degradation rates in the HEPES buffer were slower than I expected them to be, so the buffer solution was switched to MOPS, M-O-P-S buffer in the second part of the experiment. After creation of samples of known Ptaquiloside, buffer, and metal cation concentrations, with the exception of the control set in the glove box, I pipetted the solutions into about 270 separate 2 milliliter HPLC vials and crimp sealed each by hand. I think I definitely had some arm muscles going on at this point. Vials were stored at room temperature away from light. Each vial was wrapped in aluminum foil to prevent exposure of light to the Ptaquiloside during storage, as light is also known to increase the degradation rate of the compound. Every day, when possible, I tested samples in duplicate in a high-pressure liquid chromatography machine, HPLC, to determine the Ptaquiloside content of the vials. To explain a little, HPLC is a method that you can use that involves injecting a dilute solution of a material you are interested in into a pressurized machine filled with a solvent. In this case, we're using a mix of acetonitrile and triple deionized water as our solvent. The solution and solvent passes through what's called a column, which separates materials in the solution based on size and polarity of the molecules. Bigger molecules are molecules that stick better, so the column contents pass through slower than smaller and less sticky molecules. At the end of the column, a reader, in this case using UV light, detects when molecules pass by and graphs these times and magnitudes as peaks on chromatograms. You can then use the information you have gained from the chromatograms to calculate how much of your study substance you have. In this case, you can find Ptaquiloside coming out of the column after about a minute. The machine has to continue to run at different solvent gradients after this, though, to ensure the column is cleaned out of any molecules that may have been left behind before you start analyzing your next sample. Each value took about 15 minutes to analyze, top of the time needed to analyze a standard and make a standard curve, which is very important to do each day, as slight changes in pressure, temperature, etc. in the machine from day to day can change output of the machine by slight amounts. Each analysis period took about 5 hours to complete in a day. At the end of running several hundred samples over the course of 2 months, with some hiccups, I was finally able to graph my results, and I found little to no effect. A little bit of a downer for my first major research project, but not necessarily a bad result when you really think about it. Because what does no effect really mean? It means that there's one less issue to think about when trying to figure out where Ptaquiloside intrusion in groundwater may be a problem. As stated before, we know that temperature, pH, microbial organisms, and clay content have some effect on Ptaquiloside stability, but now we know that we may not have to worry about cation content of water when looking at Ptaquiloside availability on a larger scale, such as when you're trying to make geographic models of areas of possible concern. It decreases the number of variables you have to think about. I'm not letting my research stop here, though. The big question of whether or not Ptaquiloside may be a problem in groundwater is still out there. So I will be returning to the University of Copenhagen this summer, 2015, on an even more awesome project funded by my friends in Copenhagen and hopefully the Center for Undergraduate Research and Fellowships here at the University of Pennsylvania. I still have to hear back from Penn, so fingers crossed. This new project, devised by Professor Hans Christian Bruun Hansen and I, seeks to find a more direct answer to the groundwater question. This time, we'll be conducting actual groundwater samples from all over the country of Denmark and watching the degradation of Ptaquiloside in actual groundwater types. We'll be testing the water for all sorts of components like chemical content, metal cations, mineral content, conductivity, etc., and then seeing if any of these has a profound effect on Ptaquiloside degradation. It's also great because Danish aquifers do vary quite a bit on groundwater content, and the data from these experiments can be used to predict what would happen in other similar water sources around the world. Anyways, I definitely say that my research taught me tons of lab and research skills that I would have never otherwise had the chance to learn, and the people at the Department of Plant and Environmental Sciences at the University of Copenhagen are really awesome people, and they are the absolutely best for giving me this opportunity and helping me out along the way. I will be very excited to be going back. If any students at Penn or other schools want to contact me about getting involved in research, where to start, or was like doing research in a field I didn't know too much about beforehand, I'll try to answer any questions I can. You can reach me at my school email at wsiyuan@sas.upenn.edu. And thank you for listening to my first Ben Talk submission. Hopefully I'll be able to give you all an update on my research this upcoming summer around this time next year.